Hi, today I'm going to be reading an article by Dr. Ray Peat, and it is called Tissue-Bound Estrogen in Aging. And can be found on his website, raypeat.com, in the article section. This is how it goes. The quote-unquote estrogen replacement industry is based on the doctrine that a woman's tissues are depleted of estrogen after menopause. This doctrine is false. The concentration of a hormone in the blood doesn't directly represent the concentration in the various organs. The amount of estrogen in tissue is decreased when progesterone is abundant. In the absence of progesterone, tissues retain estrogen even when there is little estrogen circulating in the blood. Many things suggest an increased estrogenic activity at menopause. For example, melatonin decreases sharply at puberty when estrogen increases, and then it decreases again at menopause. Prolactin, stimulated by estrogen, increases around puberty, and instead of decreasing at menopause, it often increases. And its increase is associated with osteoporosis and other age-related symptoms. Estrogen is produced in many tissues by the enzyme aromatase, even in the breast and endometrium, although these are considered quote-unquote target tissues rather than endocrine glands. Aromatase increases with aging. Estrogen is inactivated mainly in the liver and brain by being made water-soluble by the attachment of glucuronic acid and/or sulfuric acid. Estrogen's concentration in a particular tissue depends on many things, including its affinity or binding strength for components of that tissue, relative to its affinity for the blood. The activity in that tissue of the aromatase enzyme, which converts androgens to estrogen, the activity of the glucuronidase enzyme that converts water-soluble estrogen glucuronides into the oil-soluble active forms of estrogen, and the sulfatases and several other enzymes that modify the activity and solubility of the estrogens. The quote-unquote estrogen receptors, proteins which bind estrogens in cells, are inactivated by progesterone and activated by many physical and chemical conditions. Inflammation activates beta-glucuronidase, and anti-inflammatory substances, such as aspirin, reduce many of estrogen's effects. Doctrines are admitted into the quote-unquote scientific canon by those who have the power of censorship. In astronomy, Halton Arp's discovery of quote-unquote anomalous galactic red shifts is practically unknown, because the journal editors say the observations are quote-unquote just anomalies, or that the theories which could explain them are unconventional. But the actual problem is that they are strong evidence against the Big Bang, Hubble's law, and the expanding universe. American science since the 1940s has probably been the most censored and doctrinaire in the world. Gilbert Ling's revolution in cell biology remains outside the canon, despite the profound influence of MRI, which grew directly out of his view of the cell, because his work provided conclusive evidence that cells are not regulated by, quote-unquote, semi-permeable membranes and membrane pumps. Every field of science is ruled by a doctrinaire establishment. Charles E. Brown Sequard, from 1817 to 94, was a physiologist who pioneered scientific endocrinology, but who was ridiculed because of his claim that extracts of animal glands had an invigorating effect when injected. His place in the scientific canon is mainly as an object of ridicule, and the details of his case are perfectly representative of the way our quote-unquote canon has been constructed. The argument for dismissing his observations was that he used a water extract of testicles, and, according to the 20th century American biologists, testosterone is not water soluble. And so the water extract would have, quote unquote, contained no hormone. The argument is foolish because living organs contain innumerable substances that will solubilize oily molecules, but also because Brown-Séquard was describing an effect that wasn't necessarily limited to a single chemical substance. The transplanting of living cells to repair tissues is finally being accepted, but the pioneer in promoting tissue regeneration or repair with the transplantation of living, dead, or stressed cells, V. Filatov, L.V. Polzev, W.T. Summerlin, for example, were simply written out of history. And I'm sorry that I did not pronounce those names correctly, most likely. If Brown-Séquard's extract couldn't work because testosterone isn't soluble in water, then what are we to think of the thousands of medical publications that talk about quote-unquote free hormones as the only active hormones? Quote-unquote free hormone is defined as the hormone that isn't bound to a transporting protein, with a more or less explicit idea that it is dissolved in the water of the plasma or extracellular fluid. Brown-Séquard's tissue extracts would have contained solubilizing substances including proteins and phospholipids, so the oily hormones would certainly be present and active in his extracts. But the thousands of people who ridiculed him committed themselves to the fact that steroid hormones are insoluble in water. By their own standard, they are selling an impossibility when they do calculations to reveal the amount of quote-unquote free hormone as something distinct from the protein-bound hormone in the patient's blood. The immense hormone replacement therapy industry, which Brown-Séquard's experiments foreshadowed, is based on the fact that the concentrations of some hormones in the blood serum decrease with aging. At first, it was assumed that the amount of the hormone in the blood corresponded to the effectiveness of that hormone. Whatever was in the blood was being delivered to the quote-unquote target tissues. But as the idea of measuring quote-unquote protein-bound iodine, or PBI, to determine thyroid function came into disrepute because it never had a scientific basis at all, new ideas of measuring quote-unquote active hormones came into the marketplace, and currently the doctrine is that the quote-unquote bound hormones are inactive, and the active hormones are quote-unquote free. The free hormones are supposed to be the only ones that can get into the cells to deliver their signals, but the problem is that free hormones exist only in the imagination of people who interpret certain lab tests, as I discussed in the newsletter on thyroid tests from May of 2000. In the 1960s and 1970s, when the PBI test was disappearing, there was intense interest in, a kind of mania regarding, the role of quote-unquote membranes in regulating cell functions, and the membrane was still seen by most biologists as the semi-permeable membrane, which, quote-unquote obviously, would exclude molecules as large as albumin and the other proteins that carry thyroid and other hormones in the blood. In reality, and experimental observations, albumin and other proteins enter cells more or less freely, depending on prevailing conditions. The membrane doctrine led directly to the quote-unquote free hormone doctrine. The issue of arguing about which form of a hormone is the quote-unquote active form has to do with explaining how much of the blood-carried hormone is going to get into the quote-unquote target tissues. If the membrane is a quote-unquote semi-permeable barrier to molecules such as hormones, then specific receptors and transporters will be needed. If the concentration of a hormone inside the cell is higher than that in the blood, a quote-unquote pump will usually be invoked to produce an quote-unquote active transport of the hormone against its concentration gradient. But if the membrane regulates the passage of hormones from blood to tissue cells, And especially if pumps are needed to move the hormone into the cell, how relevant is the measurement of hormones in the blood? Within the blood, progesterone and thyroid hormone, T3, are much more concentrated in the red blood cells than in the serum, since it isn't likely that red blood cells are quote-unquote targets for the sex hormones or for progesterone or even thyroid. Their concentration, quote-unquote, against their gradient in these cells suggests that a simple distribution by solubility is involved. Oily substances just naturally tend to concentrate inside cells because of their insolubility in the watery environment of the plasma and extracellular fluid. Proteins that have quote-unquote oily regions effectively bind oily molecules, such as fats and steroids. Even red blood cells have such proteins. In the case of oil-soluble molecules such as progesterone and estrogen, it's important to explain that most of their quote-unquote binding to proteins or other oil-loving molecules is really the nearly passive consequence of the molecules being forced away from the watery phase. They are hydrophobic, and although it would take a great amount of energy to make these insoluble substances enter the watery phase, the attractive force between them and the cell is usually small. This means that they can be freely mobile while quote-unquote bound, or concentrated within the cell. The oxygen atoms, and especially the phenolic group of estrogen, slightly reduce the hormone's affinity for simple oils, but they interact with other polar or aromatic groups, giving estrogen the ability to bind more strongly and specifically with some proteins and other molecules. Enzymes which catalyze estrogen's oxidation reduction actions are among the specific estrogen binding proteins. Many proteins and lipoproteins bind steroids, but some intracellular proteins bind them so strongly that they have been, in a very teleological, if not anthropomorphic way, considered as the switch by which the hormone turns on the cellular response. In the popular doctrine of the estrogen receptor, a few molecules of estrogen bind to the receptors, which carry them to the nucleus of the cell, where the activated receptors turn on the genes in charge of the female response, or the male response, or the growth response, or the atrophy response, or whatever genetic response estrogen is producing. Once the switch has been thrown, the estrogen molecules have fulfilled their hormonal duty and must get lost, so that the response isn't perpetuated indefinitely by a few molecules. Although the estrogen receptor doctrine is worse than silly, there are real proteins which bind estrogen, and some of these are called receptors. The uterus, brain, and breast, which are very responsive to estrogen, bind or concentrate estrogen molecules. When I was working on my dissertation, I tried to extract estrogens from hamster uteri, but the chemical techniques I was using to measure estrogen weren't accurate for such small quantities. A few years later, S. Batra was able to extract the estrogen from human tissue in quantities large enough for accurate analysis by radioimmunoassay. That's from Batra 1976. His crucial observation was that the difference in estrogen concentration between tissue and blood was lowest in the luteal phase when progesterone is high. Here's a quote. The tissue/plasma ratio of E2, or estradiol, ranged from 1.45 to 20.36, with very high values in early follicular phase and the lowest in mid-luteal phase. This means that progesterone prevents the tissue from concentrating estrogen. He made similar observations during pregnancy, with tissue estrogen decreasing as blood progesterone increase, so that there is less estrogen in the tissue than in the plasma. But in women who aren't pregnant and when their progesterone is low, the tissues may contain 20 to 30 times more estrogen than the plasma in equal volumes. In aging, the sharply decreased progesterone production creates a situation resembling the follicular phase of the menstrual cycle, allowing tissues to concentrate estrogen even when the serum estrogen may be low. In postmenopausal women, the tissue concentration of E2 was not significantly lower than in menstruating women in follicular phase. That was from Ackerlund et al. 1981. Besides the relatively direct actions of progesterone on the estrogen receptors, keeping their concentration low, and its indirect action by preventing prolactin from stimulating the formation of estrogen receptors, there are many other processes that can increase or decrease the tissue concentration of estrogen, and many of these influences change with aging. There are two kinds of enzyme that produce estrogen. Aromatase converts male hormones into estrogen. Beta-glucuronidase converts the inactive estrogen glucuronides into active estrogen. The healthy liver inactivates practically all the estrogen that reaches it, mostly by combining it with the quote-unquote sugar acid, glucuronic acid. This makes the estrogen water-soluble, and it is quickly eliminated in the urine. But when it passes through inflamed tissue, these tissues contain large amount of beta-glucuronidase, which will remove the glucuronic acid, leaving the pure estrogen to accumulate in the tissue. Many kinds of liver impairment decrease its ability to excrete estrogen, and estrogen contributes to a variety of liver diseases. The work of the Biskynes in the 1940s showed that a dietary protein deficiency prevented the liver from detoxifying estrogen. Hypothyroidism prevents the liver from attaching glucuronic acid to estrogen, and so increases the body's retention of estrogen, which in turn impairs the thyroid gland's ability to secrete thyroid hormone. Hypothyroidism often results from nutritional protein deficiency. Although we commonly think of the ovaries as the main source of estrogen, the enzyme which make it can be found in all parts of the body. Surprisingly, in rhesus monkeys, aromatase in the arms accounts for a very large part of estrogen production. Fat and the skin are major sources of estrogen, especially in older people. The activity of aromatase increases with aging, and under the influence of prolactin, cortisol, prostaglandin, and the pituitary hormones, FSH, or follicle-stimulating hormone, and growth hormone. It is inhibited by progesterone, thyroid, aspirin, and high altitude. Aromatase can produce estrogen in fat cells, fibroblasts, smooth muscle cells, breast and uterine tissue, pancreas, liver, brain, bone, skin, etc. Its action in breast cancer, endometriosis, uterine cancer, lupus, gynecomastia, and many other diseases is especially important. Aromatase in mammary tissue appears to increase estrogen receptors and cause breast neoplasia independently of ovarian estrogen. That's from Tekmal et al. 1999. Women who have had their ovaries removed are usually told that they need to take estrogen, but animal experiments consistently show that removal of the gonads causes the tissue aromatases to increase. The loss of progesterone and ovarian androgens is probably responsible for this generalized increase in the formation of estrogen. In the brain, aromatase increases under the influence of estrogen treatment. Sulfatase is another enzyme that releases estrogen in tissues, and its activity is inhibited by antiestrogenic hormones. In at least some tissues, progesterone inhibits the release or activation of beta-glucuronidase, which according to Cristofalo and Kabakshian, 1975, increases with aging. Glucuric, or I'm not 100% on how to pronounce that, but glucuric acid, which inhibits this enzyme, is being used to treat breast cancer, and glucuronic acid also tends to inhibit the intracellular release of estrogen by beta-glucuronidase. Although there is clearly a trend towards the rational use of antiestrogenic treatments for breast cancer, in other diseases the myth of estrogen deficiency still prevents even rudimentary approaches. Ever since Lipschutz work in the 1940s, It has been established that the uninterrupted effect of a little estrogen is more harmful than larger but intermittent exposures. But after menopause, when progesterone stops its cyclic displacement of estrogen from the tissues retain large amounts of estrogen continuously. The menopause itself is produced by the prolonged exposure to estrogen beginning in puberty, in spite of the monthly protection of the progesterone produced by cycling ovaries. The unopposed action of the high concentration of tissue-bound estrogen after menopause must be even more harmful. The decline of the anti-estrogenic factors in aging, combined with the increase of pro-estrogenic factors such as cortisol and prolactin and FSH, occurs in both men and women. During the reproductive years, women's cyclic production of large amounts of progesterone probably retards their aging enough to account for their greater longevity. Childbearing also has a residual anti-estrogenic effect and is associated with increased longevity. Being aware of this pervasive increase in estrogen exposure with aging should make it possible to marshal a comprehensive set of methods for opposing that trend toward degeneration. And as usual, Dr. Ray Peat has all his references down here, which you can read through in your own time if you'd like. I always learn several new things when I read through his articles, especially around estrogen, progesterone, and aspirin. And I'm kind of doing a deep dive currently into endometriosis, and so I came across this one. But again, this is for educational purposes only, but I hope you got something out of it, and whatever research you're doing, I hope this propels you and catalyzes that, and hope that you have a fantastic day, and I'll see you next time.